Howdy, gang. I feel like I did a really crappy job there at the end describing factorial design, so I want to take my time here and just spend a few extra minutes on covering what factorial design actually is. So, suppose here that you have different interactions that you're interested in measuring. So you have two different categorical variables, and you're interested in measuring how those two variables might independently and collectively, both independently and collectively, affect your response variable. So you are interested in two or more factors that are tested simultaneously in one experiment. The great part about a factorial design, as I mentioned before, is it allows you to test for these specific interactions. So how is variable A affecting how is variable B affecting the response, and how are the effects of variable A and B combined also affecting the response. Downside to this is it's going to require much higher level of replication. Okay, so this is what we mean when experiments are fully crossed. Each replicate receives treatment applications independently and separately, and you can test for these interactive effects. So, suppose you had some sort of predator exclusion experiment. You would want to be able to exclude those predators, right? But maybe you're also interested in something else. Maybe you have a soil type as a potential covariate of interest. In that case, what you want to do is you want to have both the presence and absence of predators equally distributed across the different types of soil types. So here, the presence of predators might be something like the dashed circles, and the absence of predators might be the exclosures or the closed circles, okay, across the three different soil types, orange, blue, and gray. And notice that each one of those treatment types are equally replicated, but don't necessarily have to be right next to each other spatially, as is the case in split plot designs. Okay, now let's take a look at an example of a fully factorial design. So here's the paper that I alluded to at the end of the last video. This is a paper where Relena and I were interested in the effects of ants and soil movement, okay, on ecosystem processes like soil respiration. And so in order to evaluate that, we needed to have one set of treatments, whether ants were present or absent. So absent ant, no ants, or ants present. And we did this by replicating this experiment in a series of buckets. Just to give you an idea of how many mesocosms or bucket experiments we did in this particular paper, over a 2-year period, we did close to 200 individual buckets. So that's the level of replication that might be required in a fully factorial experiment. But not only were we interested in the presence or absence of ants on these ecosystem processes, we also wanted to do it within the context of climate change. And so in some buckets, we increased temperature, and we had ambient temperature, or so no increased temperature. So those are our two variables that we're manipulating, whether ants are present or absent, and the change in temperature, ambient temperature versus increased temperature. In order to do the fully factorial design, we had to have buckets where ants were fully absent, so no ants were present, and were conducted at ambient temperature. Buckets where ants were present, but temperature was not changed, so it was ambient temperature. Buckets where ants were absent, but temperature was changed, so this isolates the effect of just temperature. And ants are present, and temperature is also changed and increased. So this is the interactive effect of both temperature and the presence of ants on our response, which was soil respiration. What's really cool about this is we can plot each individual variable versus the response variable, okay, and see how those two different categorical treatments cross. So one of these plots that we decided to build for this particular study is called an interaction plot, okay? And in an interaction plot, you have some sort of summary statistic associated with each individual group, in this case the presence or absence of ants, and our points represent the means, the error bars represent the standard errors, okay? And wherever the two different treatments cross, where the lines of these two treatments cross with one another, that's where we're suspecting to have a significant interaction. Okay, so in this first group here, we see that there are differences between presence or absences of ants, but then at some point those two interactions cross with one another, or those two different treatments cross in a significant way. So what we're checking for here is whether or not these lines are parallel or not. That's sort of the basics of an interaction plot, and we can definitely work through some of these later on in our lab exercises. All right, there is another type of design that I didn't get a chance to talk about, and these are nested or repeated measures designs. These can be combined with other single or multiple factor designs. So a good example would be the repeated measures design, which we used in the seed dispersal experiment described in the previous video, but it was also repeated measures because we sampled and evaluated seed count at multiple time intervals. In nested designs, we have any idea in which the subsampling within each of the replicates. Now, this is when you're nesting designs, one of the big things you're trying to avoid and keep account of is pseudo-replication. Remember, we talked about pseudo-replication last week, and we want to make sure that our individual measurements within these nested designs are not pseudo-replicated. Often, this is a big pitfall for pseudo-replication. So when nesting your study design and thinking about subsampling within individual units or within individual replicates, just make sure that you are checking for pseudo-replication. In a repeated measures analysis, a good example of repeated measures analysis is the example of, let's say, evaluating hormone levels at different stages of pregnancy across these 4 women. Okay, so because you know your individual observations may not be independent of each other, right? Because you are essentially sampling an individual participant in this study at multiple time periods. So each observation is going to be influenced by the previous observation. We might employ something called a repeated measures design. Okay. Now, here we have the hormone levels collected in urine samples every 4 weeks throughout a woman's pregnancy. And what we see here is that we use these repeated measure designs whenever multiple observations on the same subject are collected at different times. So samples are not truly independent because they're coming from the same subject, but we are expecting to evaluate the change. So in this case, the change in hormone levels. The nice thing about a repeated measures design is that it incorporates temporal variability, so how a system changes across time. And you can look at variation between subjects factors and within subject factors. Multiple observations within subject factors are not independent. So this is just what I was saying before. This repeated measures design is used when we are dangerously close to breaking that rule for independence, which is necessary for our parametric statistics. Okay, so we can talk more about examples of these on Wednesday, but hopefully this gives you an idea to some additional measures and types of study designs out there. Cheers.